I'd like to show you some very simple fluorescence experiments you can do with the sky blue dye contained in Color Nature food colors. Now inside this packet of sky blue dye is a powder, and I just took that powder and dissolved it in some water to make these two blue solutions that you see here. Now the ingredients to the sky blue dye says it contains spirulina, which is a blue-green algae, and blue-green algae contains a protein called phycocyanin. And it's the phycocyanin which gives the blue color to these two solutions that you see here. Phycocyanin fluoresces red, and that's very easy to see just by taking a flashlight and shining it on the solution of phycocyanin. You can see the red fluorescence that's indicative of the phycocyanin protein. Very easy to observe. This is being done under room lights. I'm not even turning out the lights in the room. Another simple fluorescence experiment to do with a solution of phycocyanin is to show that the protein binds to metal ions. I'm just going to take a little bit of this phycocyanin solution and pour it into some test tubes here. And to one test tube, I'm going to add some copper-2 ion. And I'm going to go ahead and add that from solid copper-2 sulfate. Now on each phycocyanin protein, there is contained a phycocyanobilin moiety. And the copper-2 ion is going to bind to that phycocyanobilin moiety. And you can even see, I see a little bit of a color shift there upon the addition, just slight, upon the addition of copper-2. And what happens when metal ions bind to the protein, the fluorescence is quenched. So we can observe that by just taking our flashlight here. We'll go ahead and place our control, and you can still see the beautiful red fluorescence from the phycocyanin protein in the sample to which we did not add the metal ion. And now we'll conduct the same experiment in the sample to which we did add the metal ion. It's quite a big difference. No fluorescence. And here we do see the red fluorescence. Now if you don't have any copper-2 on hand, this experiment can be done using pennies, as long as your pennies are quite oxidized. I'll show you how we can do this. Once again, I'm going to take my solution of phycocyanin, and then I'm going to take some pennies, and these pennies, you'll notice, are quite corroded, and that's because I want pennies with quite a bit of oxidized copper on them, in the hopes that when I drop these pennies into the test tube and mix it around, that the oxidized copper will dissolve into the solution, bind to the phycocyanobilin moiety, and reduce the fluorescence of the phycocyanin protein. This is going to take quite a bit of shaking. Let's see if it works. All right. Let's do our test. Here's our control sample. Go ahead and test this for fluorescence. Sure enough, we get the nice red fluorescence from the phycocyanin protein. Let's see what happens to the sample to which we added the pennies. Not much going on there. That's a pretty big difference. So it looks like the oxidized copper from the pennies dissolved into the solution of phycocyanin and quenched the fluorescence.